Okay, good morning, everybody. Thank you, as always, for joining us for Medical Grand Rounds. And as always, we have a great presentation this week. As people join, we're going to do a couple quick updates this week, and we'll get right into it. Talia and Helena will be sending out links for CME as well shortly. We'll start with Dr. Brian Bowman, who's, of course, oversees occupational health with a couple updates first.
Thank you, Errol. And I'll just share this screen. Can folks see the screen?
Yeah.
I hope. So very quickly, guys, I'm excited about this presentation. I won't be long. I just want to give you a little situational awareness about what's going on with faculty and employees in our own health system. If you look at the total number of positive tests in employees and faculty over the last several weeks, you can see that we're still at a very low and perhaps even the lowest we've been in a long, long time. So things are looking really good there. You can see our test positivity rate, which is this dark line, has been going down of late and is very low as of right now. That is in the context of, excuse me, I'm trying to move my slide and I can't. Let's see if this will work. That is in the context, as you all know, of very disturbing trends nationally. And in California, we're sort of at a plateau, a relatively low plateau. If you look at our monthly data over the course of the entire pandemic, you can see the total number of positive tests among employees and faculty, 481 over the course of the entire pandemic. We had our biggest peak in July. You can see October still has 10 days left, but we're still at a relatively low level, and again, a relatively low percent positivity. But you know, in the context of what's going on around us, we're not an island. I won't harangue you like I do most of the times when I give these presentations, but we obviously do have to maintain our vigilance. And one of the things we need to do, folks, is we need to get our flu vaccines, vaccinations. And we are behind our pace as of last year in terms of getting flu vaccinations. There are some data issues, and you may have been asked to come in and get a vaccination when you already have. You can obviously disregard that. We're working on that. But you know, if you've been vaccinated, if you have not either been vaccinated at Stanford or sent an attestation of being vaccinated elsewhere so that we know that you have, please help us with this number right here. We still have a fairly low percentage of participation among the faculty, and encourage, of course, anybody you know to get their vaccination. And with that, I will let us move on to the main events.
Great, Dr. Bowman. Thanks so much for sharing the updates and for joining us every week. And we continue to look forward to hearing your updates as well. Thanks for emphasizing the importance of the flu vaccine. I know Dr. Mahoney and her team are sending out messages to our faculty and staff this week for anybody who hasn't gotten it. Next, we'll move on to Dr. Will Collins, who will provide some updates about census in the hospital with COVID patients.
Yeah, thanks, Errol. So I'm in my car, sorry, bit of a transportation issue this morning, but we'll move forward. So I was going to talk quickly about inpatient updates. So we can go to the next slide for me. Dr. Bowman nicely showed the graph for national trends. This is looking at trends for our 7 Bay Area counties. I kind of go back and forth which one to show, but this one breaks out the different counties. And I think just the one interesting thing, I don't know if it's signal or noise, but if you look at the purple line with the dots, that's Santa Clara County, and there is a little uptrend recently. I don't know, again, if that's signal or noise, but something to be aware of. Next slide. This is for our own Stanford healthcare system. So first showing positive cases, new positive cases by week. Again, similar to the trends for employees, the trend has come down since our peak in July. Also hospitalizations, we're still trending down from that July-August peak. So next slide. Looking at the numbers, so sorry I couldn't update the census this morning, but as of last night, we had 6 COVID positive patients with 1 in the ICU. That's at 800P, the Stanford Hospital. In aggregate, from March 1st to October 17th, we've identified 502 at Stanford. From that 502, we still have a rate of a little over 21% needing ICU care. Deaths is now at 32, which is just over 6%. Slightly more female than male. Our age range still about the same. And then we added the race ethnicity demographics here. This is 495 out of the 502 at the 800P site. And as you can see, pretty dramatic in terms of our Hispanic Latinx population. Over 50% of our patients identify as that race or ethnicity. So certainly points to the disparities there. And go to the next slide. And I'll end it there. I just wanted to thank everyone and look forward to the main presentation.
Dr. Collins, thanks as always. I know you got stuck taking your kids to daycare this morning. Opened a little early, so thanks for sticking with us from the car and joining us, and looking forward to hearing updates every week.
Yeah, thank you, Carol.
Okay, great. We'll move on now to, we're going to move on to presenting. Actually, before I do that, I want to just mention next week, we have a really great presentation, immunomodulators for the treatment of COVID-19. And our own faculty, Dr. Rotagi, Dr. Huja, and Dr. Blomkamp will be presenting on that. So looking forward to hearing from all of those doctors next week. This week, we have a great presentation talking about the Lambda Trial and COVID-19. We have Dr. Prasanna Jagannathan, who's going to be talking about that. Dr. Jagannathan is one of our own faculty as well from the Department of Immunology and Infectious Disease. He got his MD from Harvard in 2005, did his residency at UCSF, finishing in 2019, and his fellowship in infectious disease immunology in 2011, as well as a postdoctoral along the year after in 2012. He has been doing lots of different, various really exciting research along the lines of human immunology focused on the malaria-specific immune responses in pregnancy and infancy. He won the Rosencrantz Prize here at Stanford in 2008 for his work along those lines in Uganda. And this year, of course, as many of our really high-functioning faculty have pivoted to do research along the lines of COVID-19. And Dr. Jagannathan's going to talk about the research. He's one of the faculty doing research who's come out with results, as we're planning to do with a lot of our faculty here at Stanford, and also abroad. When research is completed, we're inviting a lot of them to come present, and Dr. Jagannathan is going to present on his work in the Lambda study, the interferon gamma, pegylated interferon gamma affecting COVID-19. So Dr. Jagannathan, thank you so much for joining us today. I'm going to stop sharing my slides so you can put yours up right now. And again, we appreciate you joining us to share the results of that trial. For the people who sent the messages about the flu vaccine as well, the email was sent, Dr. Mahoney, thanks for sending that. If you have any questions about that, there's an answer about where to send an email about if you have done the flu vaccine, where to send your attestation, or if you have any questions about that. So please check that out. Dr. Jagannathan, great, you have your slides up now. Thanks again for joining us, and looking forward to hearing more about this presentation.
Great, thank you, Errol. Thank you for the kind introduction, and thanks to Department of Medicine for giving me the opportunity to speak on behalf of our Lambda investigators. So what was the premise for our study? As you all know, there remains an urgent need for outpatient therapeutics for COVID-19. As you all know, most patients with COVID-19 have mild, uncomplicated disease. A very small percentage of patients progress to complicated disease. And importantly, uncomplicated infections contribute to transmission to those with comorbidities. There are several therapeutics available for patients with severe disease, including remdesivir, dexamethasone. I think we've been developing kind of good practices for patients who are critically ill in the hospital. But to date, we don't have any approved therapeutics for patients with more uncomplicated disease. Interferons are a promising group of potential anti-SARS-CoV-2 therapeutics, given their response early within the course of viral infections. So innate immune sensing of viral products leads to production of type 1 and type 3 interferons, which after binding to their host receptors, lead to stimulation of interferon-stimulating genes and a potent antiviral response. The receptors for type 1 interferons are found ubiquitously kind of throughout the body, including on immune cells. In contrast, the receptors for type 3 interferons have more restricted expression, including epithelial cells of the respiratory and the GI tract. Both type 1 and type 3 interferons have been developed for clinical applications for other indications, including other viral infections. And specifically, type 3 interferons have been tested for the development for treatment of patients with chronic hepatitis, and specifically hepatitis D. In studies comparing type 1 and type 3 interferons have been found to be better tolerated, potentially because of their more restricted receptor expression. Importantly, coronaviruses have evolved multiple mechanisms to interfere with interferon signaling. And that's actually been highlighted by some studies that have come out over the summer that have reported that SARS-CoV-2 infected patients have significantly reduced interferon signaling. So in this paper on the left, patients with severe or critical disease, shown here in purple or the red bars, had a significant reduction in interferon stimulated genes, diminished levels of circulating type 1 interferons, and diminished interferon activity. There you go. And the Peginterferon lab here at Stanford also found that SARS-CoV-2 infected patients of all disease severities had a significantly reduced type 1 interferon response to TLR stimulation among plasmacytoid dendritic cells. These and other data have suggested the possibility that interferon-based therapies may be able to enhance clearance of SARS-CoV-2 viremia, alleviate symptoms, and prevent disease progression. So supporting this hypothesis, in an earlier study from Hong Kong, published a few months back, hospitalized non-critical patients who were randomized to receive either interferon beta plus ribavirin plus lopinavir or ritonavir had significantly lower nasopharyngeal viral loads and oropharyngeal viral loads than patients given a combination of ribavirin and lopinavir or ritonavir alone. This was one of the first indications that administration of interferons could be beneficial in patient outcomes for COVID-19. And perhaps more excitingly for our study, there was a paper published in August in Nature that documented both in vitro and in vivo data supporting the use of a type 3 interferon, pegylated interferon lambda, for SARS-CoV-2. So in this figure on the left, pretreatment of human airway epithelial cells for 24 hours with pegylated interferon lambda provided a potent dose-dependent reduction in SARS-CoV-2 infectious virus production. Furthermore, administration of pegylated interferon to mice in a mouse model of SARS-CoV-2 infection was associated with prevented weight loss at 2 and 3 days post-infection and diminished SARS-CoV-2 replication in the lungs at 2 and 3 days post-infection as well. Pegylated interferon lambda had minimal effect on nasal turbinate viral titers, but virus levels throughout were limited by were close to the limit of detection. So, given these and other data, we set up the PEG interferon Lambda trial. So this was a phase 2 randomized placebo-controlled trial where 120 outpatient adults with uncomplicated COVID-19 were randomized to receive either a single subcutaneous injection of 180 micrograms of PEG interferon lambda, and from now on I'm going to just call this lambda, compared with saline placebo. Both groups also received standard supportive care. The primary outcome was the time to viral shedding cessation of SARS-CoV-2 as measured in oropharyngeal swabs. Secondary outcomes included viral load over time, time to resolution of symptoms, drug safety, and time to disease progression. We enrolled patients ages 18 to 75 years of age. We recruited both symptomatic and asymptomatic patients. If symptomatic, patients were eligible if they had the presence of mild to moderate symptoms. If asymptomatic, patients were eligible if this was their initial diagnosis of SARS-CoV-2 infection. Patients were also eligible if they had an FDA-cleared molecular diagnostic assay that was positive for SARS-CoV-2 and obtained no more than 72 hours from the initial swab to the time of commencing informed consent. Exclusion criteria included respiratory distress, so excluded participants who had a respiratory rate of greater than 20 or a room air oxygen saturation of less than 94%. We excluded individuals who had received concurrent therapy with investigational or immunomodulatory agents. And we excluded participants based on specific lab criteria, including anemia and thrombocytopenia. This is our trial schema. So patients were recruited between April and July of 2020. After informed consent and confirming eligibility, patients were randomized to receive either a single dose of Lambda or a single subcutaneous injection of saline placebo. They were followed in a pretty intense protocol. After enrollment, they were seen on 8 additional occasions from days 1 through day 28 post-randomization. Participants collected anterior nair swabs at home daily, although I'm not going to share that data within the context of this presentation. Participants also completed at-home symptom questionnaires on each day following randomization. And then when they were seen in clinic, we collected oropharyngeal swabs at these 8 time points, as well as stool samples at 3 time points post-infection. We obtained clinical and safety labs at day 0, 5, and 14. And then also have performed biobanking and immune phenotyping in our research labs at day 0, 5, 14, and 28. We have an optional portion of the study that's a long-term follow-up that I'll mention later in this talk, where we bring participants back at month 4, month 7, and month 10 post-randomization for additional assessments as well as biobanking. To conduct this study, we, under the leadership of my division chief, Upi Singh, and ID, developed the outpatient COVID clinical trial research unit in the Galvez parking lot. So in this CTRU, patients are referred from Stanford Healthcare, other clinical sites, the community, for a variety of different studies. I'll be talking about the interferon Lambda study, which was our first drug treatment trial. But now we actually have several other drug treatment trials that are either in progress or planned for the coming months. In the COVID CTRU, there's other studies that are planned, including studies of convalescent sera, vaccine studies, natural history of disease, transmission studies, as well as immunophenotyping. It's been an incredible kind of feat to set up this CTRU, and really a lot of kudos to Upi for really shepherding this process through. This shows the clinical trial tent, tent 4, for those of you familiar with the Galvez parking lot, and our first Lambda patient was enrolled on April 25th, 2020. So what can we do in the COVID CTRU? So this is a self-contained unit, has Wi-Fi, 24-hour power, security, and it's a fully functional clinical site. We have the ability to do point-of-care labs for both a complete metabolic panel as well as a CBC with differential on site. We've developed the ability to give oral, subcutaneous, and intravenous medications, and we've been open 7 days a week since April 25th, really. This is a picture of our staff wearing very cool Treat COVID at Stanford scrubs. So this is just the layout of our tent. How does this work? We have patient parking outside the tent, an intake area. This is actually this schematic has changed a little bit with the subsequent trials, but this is what we used for Lambda. We had specific stations to consent and enroll patients, provide medications, and then we have separate areas for staff seating, storage, as well as sample processing. Our point of care instruments are here in the back of the tent as well. So how do we recruit patients? We started the study at the end of April, and May, as you all know, was a fairly slow month with regards to the number of new diagnoses in the Bay Area. So we developed some fairly aggressive recruitment strategies. We developed a website, treatcovid.stanford.edu, that's now actually being used for other studies at Stanford. We developed informational kind of guidance for patients that's on that website. We developed informational videos. We have a Facebook page that we advertise, and we did get one patient that saw us on Facebook. We developed flyers in multiple languages that were circulated. But really, the main efforts to be able to recruit our patients were actually due to the heroic efforts of both UPI, who scoured patient lists and contacted patients to see if they would be interested in participating in our study, and similarly the work of Hector Bonilla, who was really critical for us to be able to reach out to the Latinx community and recruit participants who were Spanish speaking. So getting into the results, we screened 258 participants, and remarkably, most of the, a third of these participants were actually enrolled. So 136 of these participants declined participation, and 2 met exclusion criteria after screening. So we enrolled 120 participants and randomized 60 to Lambda and 60 to placebo. And of those 120 participants, 116 completed the study. 2 patients were withdrawn prematurely in both arms. We had excellent patient engagement, a 97% retention rate, and 120 patients with 9 visits each was actually a total of 1,080 visits. And among those 1,080 visits, there were less than 5% missed visits. So we felt very good about the completeness of our data set and our ability to test our hypotheses. So these are the demographics of our study. At baseline, there were no factors that were significantly different between groups. So I'll ask you to focus here, at least on the overall group. So the median age was 36. And our youngest participant was 18, and our oldest was 71. 58% of our participants were male. 63% of our participants were Latinx, which I think is really reflective of the epidemic here in the Bay Area. Around 7% of our participants were asymptomatic at baseline. So this was the definite minority of the participants. Most of our patients had some symptoms. And of those with symptoms, the median duration of symptoms prior to randomization was 5 days. This shows some additional demographics with regards to how sick the patients were. Only 10% had a temperature of greater than 99.5 degrees Fahrenheit. And this was a construct of our inclusion criteria or exclusion criteria, but the mean oxygen saturation was 98%. In terms of baseline laboratory values, the median baseline laboratory values were normal, although we did have some participants that were either leukopenic or had leukocytosis. We had some participants that were either lymphopenic, that were definitely lymphopenic, kind of consistent with what we know about COVID-19 pathogenesis. We did have some participants with elevated LFTs at admission as well, although the median was both in the normal range. After the fact, we actually went back in collaboration with Taya Wong's lab here at Stanford in the Division of Infectious Disease and evaluated seropositivity to SARS-CoV-2, the receptor binding protein, in participants at the time of recruitment. And despite participants having a median duration of symptoms of just 5 days before being randomized, we found that nearly 41% of our participants were already IgG positive at the time of enrollment. So the antibody titers were measured against receptor binding domain by ELISA, and they're seropositive if their absorbance value was greater than the mean plus 4 standard deviations of negative controls. So although this seems like a very large number, this was actually consistent with another recently not published, but some top-line data released by the Regeneron study. I'll bring that up later, which had a very similar, which showed a very similar seropositivity at enrollment. So this shows the symptom prevalence and severity at screening. So the y-axis here shows the percent of participants that reported a particular symptom. The color denotes the severity. So green is mild. This yellowish color is mild to moderate. This darker yellow color is moderate, and then you have orange to red, which is more moderate to severe. Very few patients actually reported severe symptoms. Most symptoms were mild or mild to moderate in severity. The most common symptoms at presentation were fatigue, cough, headache, body aches and myalgias, or decreased smell. And for these, nearly 50% or more of participants reported those symptoms. So this here shows the symptom duration prior to randomization. So again, shows the same symptoms here on the x-axis, and this is the symptom duration prior to randomization on the y-axis. So the median duration of all of these symptoms was 5 days. We do see a few patients with outliers that had a slightly longer duration of symptoms, but for most patients, the median duration, for most participants with symptoms, the median duration of all these symptoms was 5 days. Okay, now going directly to our primary outcome. So our primary outcome, for our primary outcome, we evaluated whether there were significant differences between arms in the time to shedding cessation, or the time to cessation of viral shedding. And as you can clearly see here in this figure, there is no significant difference observed between the two arms. The median time to shedding cessation was 7 days in both arms. Hazard ratios here above 1 would favor lambda, and hazard ratios less than 1 would favor placebo. But again, you see here that the confidence intervals are quite wide, and the p-value is of 0.29. Hazard ratios were adjusted for randomization. We stratified a randomization based on sex and age, and so those were included in our final models. But these were not different in either adjusted or unadjusted estimates. We also performed sensitivity analyses looking just, for example, at patients who were symptomatic at the time of enrollment, and we also did not, the results were similar with regards to the median days to shedding cessation as well as the hazard ratio observed. As a secondary outcome, we evaluated viral loads over time, leveraging quantitative measurements provided by the Stanford Clinical Lab. Again, we observed no significant difference in the change in viral load over time between groups. On the y-axis here, this shows viral copies per ml in oropharyngeal swabs, and this shows the study days here on the x-axis. You can see the lines are virtually overlapping with regards to viral load. The mean change in the log oropharyngeal viral load over time was 4.3 in the lambda group versus 4.9 in the placebo group, and the delta change over time was not significantly different between the groups. We next evaluated whether there was a significant difference between arms in the time to resolution of symptoms. So again, here this shows a Kaplan-Meier curve for the time to resolution of symptoms on the x-axis. The median days to symptom resolution was 8 days in the lambda arm versus 9 days in the placebo arm. Hazard ratio here of 0.94 and a p-value of 0.76. So we again observed no significant difference in the time to resolution of symptoms between groups as well. Natasha Purrington at the Quantitative Science Unit, who we worked with very closely for all of these analyses, created this figure, which shows individual symptoms here as columns. So this is chest pain and pressure, cough, runny nose, shortness of breath, sore throat, and decreased smell. Each of these different individual rows shows an individual participant, and this shows symptom days here on the x-axis. And it's stratified by patients either in the lambda arm versus the placebo arm. The colors represent the disease severity. The blue dots actually represent symptoms where we didn't assess disease severity. And what you can see just here visually is that for respiratory symptoms, so we evaluated, for example, symptom complexes, respiratory symptoms or systemic symptoms. For all the different symptom complexes or individual symptoms, we again observed no significant difference between lambda versus placebo for the resolution of symptoms. And you can see that somewhat visually here. I will just point out that for anosmia, decreased smell here on the far right, that it did seem for anosmia, participants with this particular symptom seem to have a longer duration of that symptom in comparison to the other symptoms, which seem to largely resolve by day 10 to day 14. So what about our safety outcomes? We the overall hospitalization rate in the study was 3%, and there was no significant difference in hospitalizations or ER visits between the two groups. There were 4 hospitalizations overall, 2 each in the Lambda and the placebo arm. There were 5 ED visits in the Lambda arm versus 3 ED visits in the placebo arm. Again, these were not significantly different between the two groups. The number of participants with adverse events was 25 in the Lambda arm, or 42%, versus 21 in the placebo arm, or 35%. So there was no significant difference in the number of participants with adverse events. We did, though, look for LFT-related adverse events, and we found that participants in the Lambda arm did have a significantly higher risk of LFT-related adverse events than participants in the placebo arm. This is 25% versus 8%. So another way to visualize ALT changes over time. So we measured safety labs at day 0, pre-randomization or pre-drug administration, day 5 and day 14. We observed no significant change in ALT among participants in the in the placebo arm, but in participants in the Lambda arm, there was a significant rise in ALT at day 5 that largely self-resolved by day 14. Importantly, this was somewhat expected, as similar ALT elevations have been observed in preclinical phase 1 studies of Lambda that were used before the phase 2 studies of Lambda that are currently being done in patients with hepatitis D. So to summarize, in this study of outpatients with uncomplicated COVID-19, a single injection of Lambda did not significantly reduce the time to viral clearance or resolution of symptoms compared with placebo. This is despite a plausible mechanism of action, suppression of interferon by coronaviruses, and in vitro and in vivo experiments supporting activity. Lambda was well tolerated, but associated with these self-resolving LFT increases. We feel there are several strengths of this study. This was a relatively large single-site study with a majority of patients that were Latinx and Spanish speaking, as well as male. And this was largely reflective of COVID-19 in our surrounding communities, something that's been reported to be lacking in other clinical trials and studies that have been published over the last several months. We made a significant effort to reach out to that community and make sure that our study was reflective of that community. Once patients were enrolled, we had excellent patient engagement and retention. And as I'll talk about later, we're asking patients to come back, and a majority of our patients are coming back at the 4-month visit, for example. They're really very much engaged and want to contribute to our understanding of this disease. There are a few limitations. As I mentioned in the baseline slide, 41% of our participants were antibody positive at enrollment. But again, as I said earlier, this was similar to the seroprevalence that was observed in the outpatient Regeneron monoclonal antibody study, the REGN-COVID-2 study. That top-line data for that study was released just a couple of weeks ago and showed very similar seroprevalence. That said, we have wondered whether Lambda might be efficacious earlier infection. If we had participants who were antibody negative, for example, would Lambda potentially work better in those participants? Similarly, the median CT value in our participants was 30. So one other question that we had was whether Lambda might be efficacious in patients with higher baseline viral loads, patients that might be potentially slightly sicker. We actually pre-specified analyses looking at that as exploratory analyses that I'll get to in a moment. But I guess along these points, I did want to raise some recent top-line data that was produced by another group that was just released 5 days ago. This was released from an investigator-initiated Lambda trial in outpatients in Toronto. So that study, which had 60 participants, their primary outcome was the proportion of participants randomized who were negative at day 7 post-randomization. And in that study, in the top-line data that was released, what they found was that for those participants with a baseline viral load of greater than 6 log copies per ml, 15 of 19 of those participants were negative in the Lambda group at day 7 versus 6 of 16 in the placebo group. And that was a statistically significant difference that they observed. So we clearly were interested in understanding whether or not whether antibody positivity at baseline or baseline viral load might be a significant effect modifier of our study. So to start, we first wanted to evaluate whether baseline SARS-CoV-2 seropositivity was associated with the duration of viral shedding or symptoms. And indeed, antibody positive participants had a significantly shorter duration of viral shedding compared with antibody negative participants. So the median days to shedding cessation was 10 days in antibody negative participants, shown here in red, versus 3 days in antibody positive participants, shown here in blue. IgG seropositivity was also associated with more rapid symptom resolution. I don't show that data here, but the data is very similar. So we then wanted to test whether there was effect modification between seropositivity at baseline, the treatment assignment, so that's lambda versus placebo, and our primary outcome. We indeed did find evidence of significant interaction between antibody seropositivity, treatment assignment, and the outcome, with a p-value of 0.03. However, this was in the direction opposite to what we had expected. So amongst seronegative individuals, shown here as the solid lines, lambda, which is in blue, was associated with a non-significant delay in shedding cessation compared to placebo, shown here in red. In contrast, among seropositive individuals, shown here as the dotted lines, lambda, which is in blue, was associated with more rapid shedding cessation compared to placebo. Now, these were neither statistically significant, but the interaction itself was. But again, this was an exploratory analysis. But this is not consistent with the hypothesis that lambda might be more effective in seronegative participants. It's actually suggestive of the opposite hypothesis. We also tested for effect modification between baseline viral load, treatment assignment, and the primary outcome. The p-value for interaction here was 0.15. But again, this was also in the direction opposite to what we expected. Among high viral load individuals, shown here in the solid lines, lambda, which is shown here in blue, was associated with a non-significant delay in shedding cessation compared to placebo, shown here in red. In contrast, among low viral load individuals, shown here as the dotted lines, there was no significant difference in groups. So to stratify by viral load, we just stratify by CT values. And to remind you all, CT values of less than 30 was our high viral load group, and CT values of greater than 30 was our low viral load group. So again, together, these data suggest that in this study done at Stanford in outpatients with uncomplicated COVID-19, lambda was not effective either early or late in infection or in patients with high viral loads. So what has this taught us? Or what else should we be considering? So one other possibilities to consider, given this lack of efficacy observed in this trial, include the question of whether we achieved adequate tissue levels in the respiratory epithelium with interferon lambda. Clearly, we saw the LFT increases, so we certainly appear to have achieved levels in the GI and in the liver, but there is a question of whether we achieved adequate tissue levels in the respiratory epithelium. Subcutaneous administration of pegylated interferon lambda was associated with diminished SARS-CoV-2 replication in lower but not upper respiratory epithelium in mice. And I ask you to recall that slide that I showed earlier from Dinnan et al. That was published in Nature. Some studies of type 1 interferon have evaluated inhaled routes of administration with the possibility that could be efficacious. And so one idea could be, one question could be whether type 3 interferons might be more efficacious if given by an inhaled route of administration. In addition, there is some evidence that type 1 interferon administration might be effective in hospitalized patients. So it's an unclear answer about whether lambda would be more effective in sicker patients. Alternatively, some recent papers in Science raise the question of whether type 1 interferon deficiency is more physiologically relevant than type 3 interferon deficiency. So I think there's still some question of whether, whether the in vitro findings described below really might translate to in vivo efficacy here. So despite this study not necessarily having the expected results we were hoping for when we started, I think we have set up the infrastructure to be able to do a lot of important work related to COVID-19 in the outpatient space. And I bring you back to the COVID CTRU. The Lambda trial is now completed, except for some long-term follow-up that I'll mention. But now there's been 2 new studies that have started in the treatment tent in tent 4, including favipiravir, as well as a study using the Regeneron monoclonal antibody. And there's more studies that are planned and coming online. If folks are interested in these studies or have participants that might be interested in being in these studies, we ask you to reach out to treatcovid@stanford.edu. Importantly, we've taken advantage of the touch-heavy protocol to accomplish many other things that I'd like to highlight a couple of those accomplishments here. So first, Niaz Benai's group from the Department of Pathology used samples from patients in the Lambda trial to develop an interferon gamma release assay for detection of SARS-CoV-2 T-cell responses. This figure here shows interferon gamma responses in Lambda patients, shown here in red, versus controls, and also interestingly show that interferon gamma responses actually decrease fairly dramatically between day 17 post-diagnosis and day 31 post-diagnosis. So this is equivalent to day 14 and day 28 post-randomization. Over here on the right, Tai Wang's lab evaluated whether disease severity in COVID-19 was associated with antibody determinants. And they've found some really exciting findings that in patients with severe disease, shown here in red, that these participants have significantly reduced Fc fucosylation in their SARS-CoV-2 respiratory binding domain IgG compared with participants in the Lambda trial, shown here in light blue, or in children, shown here in dark blue. And these and other data from their group suggest that these antibody determinants might be very important or as important correlates of disease severity in these patients. We've also been working with Ami Bhatt's group, and they've been studying fecal RNA shedding using samples from our Lambda trial. These figures here are stacked bar graphs representing the load of viral load in stool over time. The orange bar increases with time, which shows a decrease in percentage of patients that have detectable viral load in their stool. And they found this both for RNA. Here, this is the N1 target, genomic RNA, the N1 target, but they had similar results with the N2, the envelope, and the RDRP genomic target. But they also found that this holds true for subgenomic RNA as well, and show that, that they're able to detect both genomic and subgenomic SARS-CoV-2 RNA in these fecal samples. And so there's a lot of work that they're doing related to this to understand fecal shedding in our patients. Another study that I wanted to highlight was some studies that are being undertaken by the UTS lab. So PJ UTS's lab is evaluating the development of autoantibodies in SARS-CoV-2 patients using samples not just from the Lambda study, but from several other studies that have been supported here at Stanford. So preliminary analysis from samples from the Lambda trial suggests that about 10% of subjects have high titer autoantibodies at day 28, shown here in red, in comparison to day 0, which is shown here in gray. And at least one patient has a new IgG autoantibody at day 28 compared to day 0. This y-axis shows the mean fluorescence intensity, and the x-axis are the 16 participants that were studied thus far. So this is very exciting, and I think Dr. Utz's lab is really interested in thinking about whether or not these are actually developing as a consequence of COVID-19 infection, a hypothesis that's gaining traction, I think, in the field in terms of understanding the pathophysiology of this disease. Our group, along with Taya, Brian Greenhouse at UCSF, and Atul Butte at UCSF, is leveraging samples that have been collected from Lambda to study the innate and adaptive immune response in patients with uncomplicated COVID-19. Jason Andrews' group is studying using samples from the Lambda study to develop improved molecular diagnostics. And Karen Jacobson, an ID fellow here at Stanford. I'd mentioned Natasha Purrington, who's in quantitative science unit, and Hector Bonilla in our ID division have been studying the natural history of mild COVID-19 as well. Both within the context of this, of the first 28 days, as well as longer term follow-up. And I just want to hearken to that here. So we've now brought back patients for their first long-term follow-up visit at 4 months post-randomization. Not all participants, about half of our participants have actually made it to this 4-month mark. Of those, 59 participants have taken follow-up surveys. And interestingly, 37% of those participants report at least one symptom, with fatigue and anosmia being the most common reported symptoms. 35 of these 59 have been seen at their 4-month follow-up in-clinic visit, where we conduct a physical exam, a 6-minute walk test, a smell test, as well as biobanking. And this biobanking includes oropharyngeal swabs, anterior nares swabs, and blood. And interestingly, 43% of our participants who were seen at that follow-up visit actually did not pass this smell test, which is the identification of one or more smells. So this is really interesting data, and I think it's going to continue to provide to our understanding of the long-term consequences of COVID-19. So to end, I just want to highlight that this work really took a village, and I hope, I tried to highlight the folks that have been really instrumental in helping us to perform these studies. The kind of main Lambda group that that started back in March, thinking about the fact that we need to do outpatient studies, develop a CTRU, and get the Lambda study off the ground was really led by UPI, as well as Julie Prasanna, Jason Andrews. We couldn't have done this without our clinical research manager, Savita Kambal, who actually recently left Stanford, but was just a wonderful person to work with and helped us really get this tent off the ground. All of the data analysis was done in collaboration with the Quantitative Sciences Unit, led by Manisha Desai, but really want to highlight the work of Vidya Balasubramanian and Natasha Purrington here. Ben Pinsky in the lab performed, hundreds and hundreds of oropharyngeal swabs, and were able to get us data back on a real-time level. So really want to thank the work of the laboratory in supporting this. I had mentioned the work of Tai Wong's lab. And then, I'm not sharing any of the data that we've generated with regards to data that's been collected from biobanking. I showed some of it, but a lot of that couldn't have been possible without the support of members of my lab, shown here, as well as members of Catherine Blish's lab, who really, stopped doing what they were doing for the months of April to July, and really helped us collect samples from these participants. I want to highlight our collaborators that I mentioned before, as well as Eiger Biopharmaceuticals for providing the Lambda for the study. Lastly, thank the study participants as well as the anonymous philanthropic support that was used to support the study. So with that, I'll end and take questions.
Dr. Jagannathan, thank you so much for that wonderful presentation. I think this slide, Dr. Harrington was just saying, it takes the village, and we have a lot of comments. The impressive nature that there's definitely multiple stories here. One is the research of the Lambda study, and then perhaps the most amazing story is this village, as Dr. Harrington mentioned, to put together the CTRU, to put together this research and the collaboration and the multiple studies that are coming out of this. I want to get into some of the questions before we run out of time here, and I'll start with Kenneth Weinberg. He talks a little bit about, I'll make this question alive to everybody. The endpoints for the study seem restricted to acute infectious, particularly respiratory aspects. What is known perhaps between the degree of abnormality of endogenous IFN responses, therapy, or systemic complications like coagulopathy, cardiac neuromanifestations? He mentions long hauler. This question came up before you posted that slide on looking at more of the chronic stuff from 4 months after. Was there, and again, we only had a couple patients for me to decide who are hospitalized. I don't know if you have data along those lines about things like coagulopathy.
Yeah, so we actually, it's a good question. When we started the study, I think we didn't actually that the data kind of emerged over time. We bank samples, but some of the coagulopathy type assays, for example, D-dimers, we can't necessarily go back to and get that data. We're now collecting that data though in patients, months out. So we're actually, for example, looking at D-dimers in patients when they come back for the 4-month visit. And somewhat surprisingly, we found around 14% of our patients have elevated D-dimers 4 months post-infection. So we've just started to look at that data, trying to understand what that potentially means. And we're going to follow that longitudinally and ask the question of whether those were influenced by either the endogenous interferon response or the treatment that we gave them.
Gotcha. There's one question here, a fair question about the commenting on the use of temperature checks at many locations. I think, commenting on a low percentage of patients, at least on it, initially, I think 10% had fever. Any comment along those lines? I think we know that a lot of patients don't have temperature. We know that already. Kind of was in line with some of the stuff that we know about COVID.
Yeah, I agree. I mean, I think temperature is an early symptom. It tends to go away fairly rapidly. What's interesting is that, and also just reminding folks that these are outpatients. Often will be taking acetaminophen or ibuprofen as outpatients before they come in. So that could be another reason why we didn't see a high prevalence of elevated temperatures at their index visit. But we do have some data that I didn't show here that just actually looks at the objective temperature measured. So we gave participants thermometers and oxygen saturation monitors to take home. And so each day we asked them to take their temperature. And what we actually see is a pretty nice decline in temperatures post-randomization. It wasn't different between the groups, but we did see that temperatures normalized per se post-randomization.
Gotcha. Dr. Leibowitz asked, how did you decide between oropharyngeal, nasopharyngeal, and nasal terminate swab locations?
That's a great question. I mean, we thought long and hard and debated for days and days about the right way to do this. And then the time when we're thinking about the study back in March, we really queried whether or not participants would be willing to come back for 9 nasopharyngeal swabs. And at the time, I think we thought that oropharyngeal swabs, that they'd be better tolerated by the participants, and I think that was definitely the case. So we certainly had some participants that said they wouldn't have participated if we had done repeated nasopharyngeal swabs, and that we had where we had done, but would agree to participate if we did the oropharyngeal swabs. I think our data that there is a question because we didn't necessarily do a comparison in this particular study of OP versus nasopharyngeal swabs. The majority of our patients were positive at day 0, despite having a nasopharyngeal screening assay that was performed outside of our study. And I think the dynamics of the viral decay in our study was consistent. So we feel like our data is of high quality. With regards to the anterior nares, we performed that assay and we actually ran it for the first 10% of our participants. But given the cost of doing all of those assays, we actually decided to freeze anterior nare swabs for a secondary analysis down the road. We've actually run now a subset of those anterior nare swabs, but I think we're still trying to understand the anterior nare data at this point. But it's a good question. I think that, I think for us, we just wanted to make sure that we tried our best to make sure that the oropharyngeal swabs were taken consistently between providers that were performing the swabs, and that our data was internally consistent. And we feel quite good and strong about that.
Okay, great. Thank you. Next question. What was the size effect of the power study for, in case it was missed here?
Yeah. The study was powered to study a 50% reduction in viral shedding for Lambda compared to placebo. So with that sample size, we had a lot of assumptions. We assumed things about dropout, for example. We assumed the number of participants that would I'm gonna misspeak here, but I guess with all those assumptions, it was a fairly conservative estimate that with 120 participants that we'd be adequately powered above 80% to be able to detect a reduction of 50% in the duration of viral shedding. We did assume that the median duration of viral shedding would be 14 days in the placebo arm, and it was shorter. It was 7 days in the placebo arm. But in conversations with the QSU, they felt that despite the actual duration observed being shorter, that actually didn't negatively impact our power, that it actually might have actually increased our power to be able to detect the difference between those two groups.
Gotcha. How about pharmacokinetics? Was the dose set because the fear of more ALT toxicity?
Yeah, it's a great question. So the dose that we use, 180 micrograms subcutaneously once, is the same dose that's used in the hepatitis D trials. And so that dose was chosen based on phase 1 studies for a combination. They had developed a higher dose of 240 micrograms, but those were limited due to tolerability. And no, we're not entirely sure, but we think it's probably due to LFTs, for example. So the 180 microgram dose was actually chosen. It's the only dose that was available from Eiger, who manufactured the product. And so that was the dose that we chose. We decided to give it just as a one-time dose, largely because we hypothesized that any influence or any impact of this drug would be early within the course of infection. So we just decided to give it as a one-time dose. And the other study in Toronto had a very similar study design with the same dose as well.
And I'm just going to tack on this question just popped up by Dr. Garcia. Could a larger dose show efficacy? Why lambda rather than alpha? I don't know if you'd be able to answer those questions.
No, I think they're good questions. I think within the context of our study, we can't necessarily ask that. I do think it's an interesting question about whether, why lambda versus alpha. I think we're all excited to see the data that's coming out of Toronto and try to understand if they really did see differences in Toronto using lambda, what might underlie some of the differences between our two studies. But I do think that we're also excited about seeing data that's coming out of plant studies, for example, ACTIV-2, that are evaluating type 1 interferons as well. Because I think that it's very possible that not all interferons are created equally, and I think it could be different formulations, different administrations, different routes could lead to very different results.
Gotcha, Dr. Hujas, and our messages means all Act 3 as well, studying interferon beta as well. So yeah, definitely more info to come on that. One anonymous question here for Dr. Utz's work on the autoantibodies. I don't know if we know the answer to this as well. What are the antibodies targeted against? Probably too early to answer that question as well.
Yeah, that's the $1 million question. I think that's what's exciting. I think that they're evaluating potential hypotheses for what those are, and they actually do see they developed a Luminex array looking at multiple different parts of antibody responses against SARS-CoV-2, looking at different proteins, and so I think that they're just putting that data together to kind of really ask the question of whether or not there's some shared target between the two.
Great. And do you know if the fecal shedding with SARS, these patients, did they have GI symptoms, the ones who had the fecal shedding?
Great question. I think Ami Bhatt wants to answer that question as well. So their team is going to be looking at that. We did have a fair number of participants that had GI symptoms, including diarrhea and abdominal pain. So Ami's group is taking that analysis on.
And Dr. Jagannathan, can I ask you, you're probably the closest person, of course, to answer this question. You mentioned the top line data that came out of Toronto. It's just top line data, so we don't probably know a lot of details yet. And in this study, do you see potentially that you mentioned there might be that we didn't have sick enough patients. We're going to have more data, I think, other studies in hospitalized patients. Do you think this might be a drug that ends up being a viable treatment option down the line, or it's just too early to tell? I kind of want to put you on the spot a little bit to get your take on that.
I think it's too early to tell. I mean, I think if you had our data in isolation, I think our data, are not consistent with the idea that lambda would be effective in patients that are in outpatients with uncomplicated COVID-19, whether it was early or late in infection. So our data is not consistent with that use of lambda. I think there are other uses of lambda, that we can't necessarily comment on. The data from Toronto, I think when we see that data emerge, I think we'll need to kind of dig in and say, well, what might be different between the two populations of patients. I think, one thing for sure is that our participants are largely Latino or Latinx, and 62% of those participants. That is not the participants that they're seeing in Toronto. Their ethnic makeup is actually largely South Asian participants and African participants as well, patients of for patients of African descent. So there could be lots of, within host differences as well that I think could be worth exploring.
These are all great points. Dr. Jagannathan, again, thank you for this wonderful presentation. As we end, I'm going to ask Dr. Rogers actually had a message and had the great point of talking about that bigger story of this village, as Dr. Harrington mentioned, to put this all together. And so I'm going to ask her to help us close this out and commenting more about the CTRU and the amazing work that went here. Dr. Rogers?
Yeah, thank you so much, Errol. I just wanted to say my congratulations to you on behalf of all of us that are in the COVID clinical trials community. I think the work of your group has been really tremendous. And I just wanted to highlight for the remaining listeners just a few aspects. First, the fact that you built this outpatient infrastructure with the team that you have here in 6 weeks of seeing our first patient at Stanford, getting things through the IRB, having a protocol you believed in and could stick with is really remarkable. I think the fact that you had such outstanding patient engagement that they were willing to, drive in from home every other day for testing in your group and really stuck with it through the end also speaks to the kind of experience that they had getting care in the CTRU. And finally, just to comment on the fact that this is a negative trial, and as we all know, most negative on the top line, I guess, and as we all know, that's true of most clinical trials that we do. But I think that your group did such an amazing job of showing the reason that it's still important to do every clinical trial to learn as much as we can. And I feel like you've got amazing science coming out of this negative trial for the question, about this particular drug for our patients, but so much learned. And just for those of us that are trying to do outpatient trials, we're really standing on the shoulders of the work that you did, about the very careful phenotyping, the careful biobanking. So just wanted to congratulate all of your team on really conducting a superb trial. So thank you so much for sharing your work with us and being here today. And thank you, Errol, for the opportunity to get to say a few words.
Dr. Rogers, so much better to close on with you speaking than me. So thank you so much for that. Thank you again, everybody, for joining and watching, and everybody have a good rest of the day and week. Thanks a lot.
Thanks all.
Thank you.